Hi everyone, this is Mrs. Alexander, and this is the third and final part for our Unit 6, Chapter 7 notes. Meiosis, pronounced meiosis, is the process of making sex cells, or gametes. And pictured here are the different ways we can picture meiosis, a sperm entering an egg, sexual reproduction, and on the right actually showing how the cells divide twice instead of just once like they do in mitosis. Cell reproduction is whenever cells will actually split 2 times in their nucleus in order to make sure that each gamete or haploid cell will only contain half of the parent's DNA or chromosomes. This way, whenever they combine during fertilization, the new organism, now called a zygote, will receive half from each parent, thus restoring the diploid or full set number. To carry out reproduction, we need these sex cells. In humans or other eukaryotic cells, we call them sperm and egg. Here's a visual for the sperm meeting the egg. Once one enters, it will ward off all the other sperm, killing them, and then it will allow that newly formed zygote, which is what we call a fertilized egg cell, to continue with its mitosis process, which is the making of new body cells. Once that has happened, it will travel down the mother's fallopian tube where she will house the baby in her organs until it is time for her to give birth. This brings us to meiosis. Understand it happens after mitosis, and it's when the body wants to create sex cells. Remember, the end product of meiosis is 4 sex cells that have half of the original number. Again, we call them haploid because it has half. They are unique. They are not identical. Unlike mitosis, which produces identical body cells, meiosis is creating new original cells with only half of the chromosome as the parents. This halving of the chromosomes is where the word haploid comes from, and it happens whenever meiosis involves 2 divisions of the nucleus. That means it goes through prophase, through telophase twice, and in order to distinguish it between mitosis, we put the Roman numerals 1 and 2 after the words prophase, metaphase, anaphase, and telophase. Note, the cell only goes through interphase 1 time, and that's before mitosis or meiosis ever begins. For example, if I have 46 chromosomes in my body and I want to make a sex cell, I need to end up with half of those 46 in my sex cells, so 23. This way, whenever 2 parents contribute their halves, the new offspring only gets a total 46, which is the normal or diploid number. Having more than that, and the offspring would not be genetically correct, and it could lead to a miscarriage. Here's a quick clip comparing mitosis and meiosis.
Prior to meiosis or mitosis, DNA replication occurs. In both meiosis and mitosis, the nuclear membrane breaks down as the DNA organizes into chromosomes. In meiosis, chromosome pairs come together, or synapse, and crossing over occurs, resulting in mixing of the genetic information between the chromosome pairs. The paired chromosomes then align along the central plate of the cell and subsequently separate, one traveling to each end of the cell. In meiosis, a second division sequence occurs, resulting in 4 cells with half the number of chromosomes. Mitosis involves a single division sequence, resulting in 2 cells with no net change in the number of chromosomes.
Notice and compare meiosis on the left, you end up with 4 cells, sex cells, that have half the number. On the right, mitosis is whenever you end up with 2 cells that have the identical number of chromosomes and type. So now let's go through those phases a little bit more in depth. In meiosis 1, prophase through telophase are pretty similar to mitosis. Notice there's some of the same things happen. The nuclear envelope breaks down. You start to see the chromosomes. The main difference between mitosis and meiosis is in prophase 1, when crossing over occurs. Crossing over is important because this is what allows every individual to be unique from their parent. It allows your parent's DNA to cross over and exchange segments with each other, which gives you unique properties, not just like your mom, not just like your dad, but more like grandma and grandpa and mom and dad mixed together, all sorts of traits being exhibited at the same time. Here's a quick video on crossing over.
The cell prepares for meiosis just as it does for mitosis. But as the DNA condenses, each chromosome finds its homologous counterpart. These matching pairs of chromosomes hold equivalent genetic information, one set from mother and one from father. Next comes the first shuffle of nature's genetic deck. The two homologs trade genes in a process called crossing over. This forms new hybrid chromosomes. The spindles then place maternal and paternal chromosomes randomly on each side of midline. It is another shuffle that can lead to a host of different genetic outcomes.
From here you have metaphase 1, anaphase 1, telophase 1, and then the second time this happens we call it prophase 2, metaphase 2, anaphase 2.
Fibers pull the homologs apart. This leaves the sister chromatids intact. With twice the amount of genetic information the cells will need, nature must cut the genetic deck in two. This second division produces 4 unique sets of chromosomes. Because they carry half the genetic information, we call these haploid cells. Many of them will mature into sperm and eggs, the reproductive cells called gametes.
And no 4 sperm or eggs, gametes, are alike. They all have different DNA in them because of crossing over. So again, here are the stages in case you missed any parts. Notice the metaphase, anaphase, and telophase are pretty identical to the same steps we learned about in mitosis. There they are in picture form. One cell starts off at a diploid, the full set, and you end up with 2 that have diploid. The only difference is notice that there's a piece of the blue chromosome on a piece of the green chromosome in telophase 1. That's because crossing over occurred during prophase 1. Meiosis 2 is the second division of the nucleus, and notice that there's no difference in prophase 2. All it is really a new spindle forms. The nuclear envelope's already dissolved, you already see the chromosomes, so that's a little different. But metaphase, anaphase, and telophase is halving it a second time, so the overall finished product is 4 haploid cells that have half of the DNA. There it is in pictures for you. It shows prophase 2 through telophase 2 and cytokinesis finally occurring. Cytokinesis occurs the second time after telophase 2. How is meiosis important to our genetic variation? It's important because this allows us to have new genetic combinations. No one's the same. There are 3 ways during prophase 1 through telophase 2 that these things can happen: independent assortment, crossing over, and random fertilization. Independent assortment just means that whenever the genes are pulled apart about homologous chromosomes, they do this independently of one another. So just because you got your mom's first chromosome doesn't mean you're also going to get your dad's first chromosome if it's dominant. It also means that it's randomly like choosing Skittles out of a pile. You don't get to choose your favorite color, you just reach in a bag and grab one. Random distribution. Here is the meiosis flowchart. I want you guys to copy this down, so take a moment and press pause to do that. It shows 1 cell diploid starting out at the top. The first division results in 2 cells that are both diploid, and then the second division starts out with 2 diploid cells but ends in 4 haploid cells. So take a moment, press pause here, and fill in your flowchart. Crossing over, I'd like to get a little bit more in depth with. Again, it's whenever the DNA exchanges during prophase 1. You might be asked on the test which stage of meiosis does this occur. The first prophase, so prophase 1, and it allows part of one gene, genetic material on one chromosome to be swapped with another one, thus creating genetic combinations. Practically unlimited the way we look. That's why everybody in your family, nobody looks just alike even though they came from the same mom and dad in some cases. Furthermore, whenever a new baby forms, that's a fertilized egg, is called a zygote. The individual is created by forming 2 random sperm and eggs. So just whichever sperm gets to whichever egg. We don't have say over that unless we were to pay someone in a laboratory to choose that for us. This is the best way to create genetic variation through sexual reproduction. The pace of evolution is really quick, and it allows us to have a bunch of different organisms with a bunch of different resistances and weaknesses and strengths in our environment, and it allows all sorts of evolution to take place. In males, whenever they create their sex cells in humans, we call it spermatogenesis, and it occurs in the testicles or testes, which is the male reproductive organ, and you end up with 4 sperm every time this happens. In females, we call it oogenesis or oogenesis. The O stands for ovum or the egg. These are found in our ovaries. There are female reproductive organs, so please know that ova is the same thing as egg. Ovum is plural for ova. On the test, I might call it an ova instead of an egg. The only difference in females is that 3 of our eggs don't actually fully mature, and we call them polar bodies, whereas the one single egg gets stored in our ovaries for further use. We have talked about sexual and asexual reproduction already, but I just wanted to touch on it one more time. Asexual reproduction, you get all of the genes, you're a genetic clone. There's no sperm and egg meeting, no fusion of haploid cells like gametes. Instead, it's just a clone, and they're genetically identical. In contrast, whenever you do sexual reproduction, you have 2 parents, and you create a new individual that has half of the number your gametes do to produce an individual with the full set. Types of sexual reproduction we've talked about in class are binary fission, fragmentation, and budding. Let's take a really quick look over those. Press pause if you need to have some more time. Here's binary fission. This can be done in bacteria and amoeba. One cell gets copied into 2 identical cells. Budding is also asexual, and it's found in yeast. Notice a little bud buds off of the original organism. It's smaller than its original cell, but it grows into a big one. Same thing for Hydra. The bud is a little piece budding off. Fragmentation is whenever an organism splits itself into fragments or pieces, and each fragment becomes a new individual. That can be found in flatworms and in starfish. And sexual reproduction can be found whenever a sperm and an egg come together to create a new individual. Note that that is also called pollen in some organisms, not sperm. Sexual reproduction in amphibians and fish, note that sex can actually take place outside the body. So whenever a fish squirts its eggs into the water and another fish swims over and spreads its sperm, that is still sexual reproduction, even though no physical contact was made, because a sperm and an egg, gametes, were involved. Plants are rare and they are very confusing. Just know that they can do it sexually and asexually. Like potatoes and strawberries, they actually have these runners that will run under and produce clones for them, as well as other cells will actually use their pollen and their egg, and they can fertilize themselves or other flowers to produce genetic offspring that are unique or identical. There are disadvantages and advantages of both. Please understand that the best advantage for sexual reproduction is genetic differences. The disadvantages is the amount of time it takes and the energy it takes to grow that offspring and take care of it. Advantages and disadvantages of asexual reproduction is it's fast, and usually our strategists don't care about the quality, but the quantity and how many they can have in a short period of time. They do not care for their young, so they don't have to put all that time and effort in. The disadvantage is that clones are original to the parent, so if we find something that can kill them off, this is bad for the whole species or population. Not very much evolution occurs. Genetic diversity is important because it's what allows evolution to take place, and based on the Red Queen's hypothesis, sexual reproduction is the best form of evolution going on in today's society. Only diploid cells can repair themselves, and so that's very important to know for the evolution of sexual reproduction, and the process of meiosis is where we can have that take. Well, I hope that was a quick recap and you understood. If not, see me in class, and feel free to go back, pause, or rewind. Have a wonderful day.